Hi. Data pre-processing. So, data pre-processing are the activities to be done before actually starting the process. As a data scientist, we work with data. We collect data from the appropriate sources. We make sure that the right set of data are collected. However, we are not talking about data from some 100 entities or 100 objects. We are talking about large volume of data. Normally, data are collected from some 60,000 entities. For example, if you are going to develop a fraud management system for a bank, you will have to collect from at least some 100,000 customers. So, whoever applied for a loan in the past few years, some 100,000 records you will have to capture, and then you will have to do the analysis. So, when we are dealing with such large volume of data, quite often the problem is there may be some issues in the data, there may be some mistakes in the data, there may be some gap in the data, wrong data has been entered. So it is the responsibility of the data scientist to make sure that you are not misled because of the error in the data. That is why we are doing pre-processing, data pre-processing. After collecting the data, we do some number crunching to make sure that there is no mistake, there is no gap. That is called data pre-processing activities. So, what is the data pre-processing? Any type of pre-processing activities performed on the data, performed on the raw data, because whatever we collected, we call it raw data. It's a raw data, so that it is ready for the data science project. So you collect the raw data, look at the picture, you collect the raw data. Pre-processing is an activity which takes raw data as an input and refined data as the output. So this is called data pre-processing. There are various steps in pre-processing. The first activity is called data discovery or data profiling. See, once the data is collected, you will have to discover the content of the data. You will have to discover the meaning of the data and profile them. That is called data discovery and profiling. Then there are some cleansing activities required. Is there any mistake? Is there anything? And so those type of things. Simple thing is, when I do some research, you just pick up all the 60,000 records of data, write a small Python code to create a pandas object and find out the minimum, maximum, mean, standard deviation for each column. For a particular column, if the minimum happens to be 0, what does it mean? There is a mistake. For example, age of the person, if it is 0, then it means that there is a mistake. The blood pressure of a patient, so the minimum happens to be 0. It means there are at least one row with 0 entity. Now you can think of correcting that. So these type of activities. Then The third activity is called data reduction. Optimization of the number of columns and other things. Removing unwanted rows, removing unwanted columns. Then we do an intelligent activity called data transformation and enrichment. So transformation, enrichment. After doing all these things, we do a set of validations to make sure that we are having the right quality of data. And then we publish. So why do we do this data pre-processing activity? Because we want to clean up the data and increase the quality of data. Because the whole of the data science activity depends upon the quality of the data. Quality of the result depends upon the quality of the data. Quality of the prediction, quality of the and recommendation for the business depends upon the quality of the data. That is why we take enormous amount of steps to make sure that the data pre-processing is done with the highest quality. Again, let us repeat what are the key steps. There are 6 key steps in data processing: data discovery and profiling, data cleansing, data reduction, data transformation, enrichment, and validation and publishing. Let us elaborately see data discovery and profiling. So here, as a data scientist, one of the important quality for the data scientist is to understand the domain in which you are doing research. So it's important for the data scientist to look at the data, understand the meaning of each of the column, understand the range of each of the column, what exactly is the value. So is it correct? So that is what is called discovery and profiling. So what you should do is, you should collect statistics about its quality. For example, if there is a column called body mass index and there are totally 15,000 rows, verify if there are any zero entities. What is the body mass index range? You try to understand from the domain expertise. See whether they are all in the same order. Try to find out if you can find out the mean of the column, find out the minimum, find out the range. If the minimum happens to be 0, it means for some of the records, the body mass index is 0. Find out how many have zeros. Check how to correct it. So it is about understanding the meaning of each of the column, each of the value, and make sure that they are corrected. So what is it? Collect statistics about its quality. Data profiling or data discovery and profiling is the process of examining, analyzing, and reviewing the data to collect statistics about its quality. So what is the percentage of body mass index above a particular range? What is the percentage of body mass index between a particular range, the mean minus delta plus delta? So you find out all those things and make sure that the quality of the data is right. So that is called data discovery and profiling. In data discovery, there are several approaches, several techniques. One is called column profiling. Column profiling is about choosing each column in the data, collecting the statistics, understanding with the domain expert, and make sure that we have the right quality. If there are any gap, we will have to correct it. That is called column profiling. If a particular column is about the height of the person in centimeters together with decimal points, then make sure that you understand what is the height of a normal person, so that the minimum, maximum range, all these things will fall in place. That is called column profiling. Column profiling assesses tables and quantifies the entries in each column. Then the second is about cross-column profiling. Cross-column profiling is about comparing two columns, dependency analysis. For example, you have a body mass index, you have a glucose level. Now you compare them. The first one was about simply column profiling. Here you see whether they are correlated to each other. In real life, if there is a positive correlation, then column 1 increases if column 2 increases. So, for example, in mark processing, the mark of mathematics and science may have correlation. There are certain places where they will be negatively correlated. If one value increases, another value will decrease. So these type of analysis has to be made. That is called cross-column profiling. Cross-column profiling features both key analysis. So there are two keys. So you analyze and you do some dependency analysis to make sure that the data is of high quality. Then the third technique is called cross-table profiling. Here there are multiple tables involved. So cross-table profiling uses key analysis to identify stray data as well as semantic and syntactic discrepancies. So across the table, if you find out some stray values, you can find out. Similarly, data rule validation. For example, in a particular domain, if there are certain data validation rules, you please apply. In the marks, for example, if one student is a bright student in mathematics, his other marks must also be higher. Similarly, in medical data, there can be some validations. If the particular reading is high, then another particular reading must be low, possible. So these type of data validations can be done. So what is data discovery and profiling? Data discovery and profiling is more about understanding the data, understanding each column, understanding cross column, understanding across the table, understanding the rules, data validation rules, and make sure that mistakes are not there. So that is called data discovery and profiling. The second is called cleansing. Cleansing. Sometimes one record will be completely zero. Sometimes one record will have some high values. Sometimes instead of typing 1 0, he might have typed 2 more zeros. So data cleansing ensures that raw data is corrected, cleaned, and it is suitable for feature engineering. So rectify the quality issues such as bad data are eliminated. So if the age of the person is 82, when typing 82, supposing he types 8222 because of the mistake in pressing the key, the person's age will be shown as 8222, which is a bad data. Sometimes while collecting data, some of the data may be missing. In the patients, some test might not have been done by somebody, so that will be missing. So identify and sort out the missing data, reduce the noisy data, identify and remove duplicates. Sometimes duplicate entries may be there. So this is called data cleansing. Data profiling is about understand the meaning of each of the column, each of the value, and so on, and try to adjust them, correct them. Whereas data cleansing is simply about numbers. Find out whether any bad data are there, any mistakes are there, anything is missing, anything is a duplicate. Remove all of them. That is called data cleansing. The third one is called data reductions. Usually there will be redundant data. For example, column row redundancy is 1. It means for the same patient there may be multiple records. For the same patient, for the same individual, there will be multiple records. It's possible. So that is called data duplication. So data duplication will be removed. That activity is called data deduplication. Similarly, there is an activity called cross-volume deduplication. The ability to remove across application volumes. It's not individual rows, individual application wise sometimes a duplication may be there. That can be removed. Data compression. Data compression is about making the data simple and clean and independent rather than a complex form of data. Similarly, small file optimization is another approach. So in data reduction, we are removing redundant data. The redundancy may be in the form of row or column. So removing those are called data deduplication. If the date of birth is there, age need not be there. If the date of joining the company is there, then experience need not be there. So these are all duplicate entries. We can retain only one of the column, and the other can be removed. Similarly, data can be put up in a simple form rather than complex form. That is called data compression. Small file optimization is another interesting approach. Data transformation and data enrichment. This is organizing the data to make the most sense for our goal. From a domain expertise perspective, you understand the problem. You are the data scientist. You understand the problem. You understand exactly what you are going to do. So in such a case, what we are going to do is, we want to put them in the right form. There may be unstructured data available. You may want to structure them into structured data. You may want to combine salient variables when it makes sense. For our particular type of study, some of the data may not be required, and we may want to combine certain variables. Similarly, we can do the data normalization. What is data normalization? Normally, when you are collecting the data for particular entities, for example, in the medical data, you may collect the height of the person, weight of the person, body mass index. All these things are selected. We know what is the average height of the person, what is the approximate weight of the person, what is the approximate body mass index. All these things are known. However, when you want to know the number of red blood cells, RBC count, WBC count, the number is too big. This is here, something is 48, there it is 1,20,000, 1,80,000. So one column has numbers in the range of a few million. Another column has a number in the form of 150, 180, 170. Now, when you create a model, the calculations are not coming up correctly. That is why, if you want the model to be more accurate, all the data can be normalized into a certain range. Usually, what we do is, for each column, you find out the mean, you find out the standard deviation. Each data x equal to x minus mu by sigma. If you subtract by mu and divide by sigma, you will get all the numbers between -1 and +1. So what you do is, in body mass index, you find out the mean, you find out the standard deviation. Then for each entry, mu is the mean, sigma is the standard deviation. For each entry, you change into x minus mu by sigma. Now the entire column will become numbers between -1 and +1. Similarly, if you do the same thing in RBC count, WBC count, in RBC count, you find the mean, you find the standard deviation. For each RBC count, you now say minus mu by sigma. If you do this, RBC count also, all the entities will become -1 to +1. This is called normalization. Similarly, WBC count. So that now we have all the data, each of the column is between -1 and +1. Now if you build the model, they are more accurate. Of course, finally, when you are interpreting the result, you will have to do certain things. So that is called data normalization. Data validation and publishing. Data validation consists of validating the consistency. Again, are they consistent across the table, across the columns, across the rows? You can find out. You can find out the completeness. If one column has 15,725 entries, every column must have 15,725 entries. Accuracy. So all these things are calculated. Then we should also divide them into training data and testing data. Usually they are divided in a homogeneous manner. So all sorts of combinations must be there. So make sure that testing data is a true representative of the training data as well. So from the whole data, 80% of them are segregated as training data and 20% of the data are segregated as the testing data. Make sure that all the homogeneous things, they are heterogeneous actually, heterogeneous. All possible combinations must be in the training data and all possible combinations must be in the testing data. That is called data validation and publishing. So, in this lecture, what have we studied? We have studied about data pre-processing. So, data pre-processing is essential because data science is all about the study of the data. Unless the quality of the data is high, your result may not be high. Your result may not be reliable. That is why we do data pre-processing. There are various types of various steps involved. One is data discovery and profiling, cleaning, reduction, transformation, and validation and publishing. So these are the steps involved in that. We have seen elaborately about what is normalization that is also required. Thank you very much.